So transcription is when DNA is initially bound to a RNA polymerase, and then after that, it is elongated and then read and transcribed into a mRNA compound, or mRNA, which is then later translated. To show this transcription process, though, I'm going to go ahead and just simplify the DNA strand. I hope you can see it right here. So this is going to be what we call the template. This is the template part of the DNA right here. So what that essentially is going to be what the RNA polymerase copies itself off of. So what happens is in this region, which is the promotional region, there's 3 areas of it. Ultimately, you have the core region, which is directly in front of the TAT or TATA box, or the actual gene that we're going to be transcribing. The most distal region, oftentimes, or it's referred to as the -35 region, is one of the main influencers and regulates the actual transcription. And then this central region, I don't know what that symbol is where most of the bonding happens, but this is going to be the promoter region, which is at the very beginning of the DNA gene that we're going to read and transcribe. So this template gene is therefore a RNA polymerase. We're just going to call it RNAP. This little guy right here is going to go ahead and initiate the process and read, or go ahead and begin to read the RNA, or the DNA, to make mRNA. So what happens is the RNA polymerase bonds initially to this promoter region and begins to unzip the DNA and its bonds that it has, or its little chain linkages here, the bases of the codons, and the codon bases. And it begins, and from here, it reads, so like, let's say, let's go ahead and just, start codon. It will read a start codon as the first one, and that'll be the initial thing. So it's always going to start with a codon triplet of AUG, which is methanine. So this first little line, which is going to represent one of our codon groupings, is going to be AUG, which is a start codon. And what this tells the RNA polymerase is that it's going to have to use complementary base pairing, which is essentially the idea that there will always exclusively be a hydrogen bond between the 4 variations of codons. So there is technically 5. There's the A, G, C, T, and U, but that one's more for RNA. A will always have a hydrogen bond with T, and G and C will always hydrogen bond with each other. So, for this start codon, the RNA polymerase is going to go ahead and write out a T, or it would write out a T, but since DNA and RNA aren't the same, it's actually going to replace the it's going to replace it with uracil. So instead of having a T for the A in its complementary base pairing, it's going to be a U. So it would end up starting out as a, yeah, it would read out like this. I gave myself a tizzy. So this start codon will actually be produced by the RNA polymerase into the messenger RNA form. This little, so as this guy goes and absorbs this region and transcribes along this direction and reads that start codon that's right there, it's going to translate it into its mRNA form, messenger RNA, which is the RNA that leaves the nucleus in the end, because this is all happening inside of it, is going to start with a U, A, C, and go on and on and on and on and on and on. Because of that complementary base pairing. It will always have the opposite of what it pairs with. So C to, or the T to the A, and the A to the T, and the C to the G, and the G to the C, and replace T with U in RNA sequences, not DNA sequences. Ta-da! So now that this whole process has started, and it's elongated, unzipped, and it's read the start codon, and it's started to actually put in a genetic code, or a series of codons, until it reads a stop codon, which will allow for the RNA, or the messenger RNA, so this little strand that's going to be produced out of the RNA polymerase right here, it is going to, I had a brain fart. It'll go ahead and once it reads that stop codon, it'll be released from the polymerase, or the RNA polymerase, and then the messenger RNA will actually be able to leave the nucleus and begin its translation by ribosomes. Prokaryotic cells do it a little bit differently. So when the RNA needs to be released, because there is no nucleus, it's all in the same little thing. What will actually happen instead is there is a there is a protein that is attached to the RNA polymerase that binds and actually closes off the RNA polymerase, stopping its ability to transcribe for that period of time. Next is translation. Translation is the steps after the messenger RNA has left the nucleus. So we're going to go ahead and start with our messenger RNA and its codon triplets that it's got right here. Now what happens is when this mRNA leaves the nucleus, it's going to go ahead and be surrounded, essentially, by ribosomes, which are made out of rRNA, which is the ribosomal version of the RNA codon strain. What this does is it allows for the creation, or it allows for the ribosome to establish its small subunits as well as its large subunit. So it's kind of upside down right now, but we're going to work with it. So what happens is in these, once this ribosome attaches to the mRNA, it's going to go ahead and automatically read this first codon right here, which is going to be our start codon, which is AUG. And that is going to call in and essentially get a tRNA to come in and bring it. Let me draw a little tRNA real quick. A tRNA, which is going to be right here, also has a very similar, it's not a codon, but an anticodon base, which is going to have, you guessed it, based off of the complementary base pairing law of DNA and RNA, is going to associate with the opposing or paired codons that it has. So this anticodon would have the resolving, the other side, or the coding side of the DNA that we missed out, that wasn't translated or transcribed into the mRNA here. These anticodons are always going to be attached to an amino acid, which ends up forming the polypeptide chain and the protein that we're trying to produce in this. There are three main sites in the ribosome that happens, or where the actual process of translation is. There is the entrance site, or the acceptor region, which is going to be the A region. Then there is the P region, which is where the polypeptide chaining begins. It's upside down. Whoopsies. P. My camera? There we go. And then last but not least, we have the exit region, E. So the acceptor region is the region that comes, or where the tRNA will go ahead and fly in, as long as it has the anticodon to match or to be anti of the codon that is being read by the ribosome. It'll come in to this acceptor site. This ribosome will shift and put the polypeptide site, once the polypeptide chain from the tRNA before it is started, it will enter this site and it will drop its, or it will have its polypeptide, or it will begin the chaining. The amino acid, this little guy up here, this little guy right here, is going to be chained together at this exact region and site right here. This is where the bonding and the chaining of the polypeptide amino acid chain is. So after it goes to the acceptor site and it goes and it has its polypeptide and it hits the peptide region and drops its amino acid, or has the ribosome essentially hold on to it and begin the bonding between them, it goes to the exit site where the tRNA then leaves to go be elsewhere. It's no longer useful. It doesn't have the amino acid that we wanted it for. As it continues, as this process continues onwards, and as more and more tRNAs come and pair with the mRNA in the P region, more and more amino acids are going to be added on, forming a long chain-like structure right here. Now, what finally happens when all of this is during this process where this chaining and everything happens, and while the ribosomes are surrounding the mRNA is made longer. It's stretched out because it's tense and tight and smushed together due to the tight codons and the bonds that are formed there. With this, finally, that is really the center aspect of translation. Finally, we are going to, or the mRNA is going to have a stop codon sequence, probably, at the end here. That was also coded in by the, that was in the DNA sequence that it transcribed earlier. This is either going to be a UAG, a UAA, or a UGA. And this will actually cause, any one of these three will cause the ribosome right here to release and detach, so the polypeptide chain can leave and go on and perform its actual duty. and that would be the termination of the translation of the mRNA into a polypeptide chain of amino acids through the usage of ribosomes and tRNA, or